I don't know how to do that. And I was like, oh, even though
So you can't split.
Okay, so a few announcements. So remember, we have our next exam on Wednesday. So I know that's come up really quick. It's going to be a little shorter exam, and I'm planning on lecturing the last 15 minutes. So I'm going to give you 55 minutes and, allow you to take a quick break if you need it, and then last 15 minutes. We're a little bit behind where I was hoping to be, and that's kind of why. And because the exam 3 and exam 4 were pretty close together, I just made it a little bit shorter exam. So I think there's 52 questions, something like that. So a few more than what that first exam that we had was, but okay, so that's going to be covering chapter 15, which is a really short chapter, the epidemiology, the innate immunity, and the adaptive immunity, that I finished by posting that 15-minute lecture on Wednesday. Okay, so we're going to take some time to review a little bit today. So I've got some practice questions. There was a review question that I ended that recorded lecture with, and we'll get to that as well. Okay, so we'll start with that. I am available tomorrow morning when I'm usually not. So I'll be in my office all morning tomorrow, 8:30 to noon. I have lab from 12 to 2. And then if somebody would like to see me after that, I just let me know. So I'm around, but I'm available after 2 as well. Wednesday, I am not very available because I'm teaching someone else's lab at 8. I have my own lab at 10. So I'm really only available like the hour or so before the exam on Wednesday. Okay. Okay. So any other questions? All right, so I've got maybe 5 or 6 practice questions here, and then we'll go on to talk about vaccines today, which will not be on Wednesday's exam. Okay, so that's section 17.5. So again, homework from 15, 16, and 17, except 17.5 I pushed off. Okay. All right, membrane attack complexes, complement. I'm having a number of questions, those of you that are coming to office hours, that want to talk about complements. So take a second if you haven't decided which of these is your best answer. We'll talk about it. We'll review complement a little bit. So you can see by one multiple choice question, you can kind of get at a lot of different things. Okay, correct answer here should be D. Okay, so those membrane attack complex, actually answer E is really close to being correct. If you didn't know all your numbers, the membrane attacks are C5, C6, C7, C8, and C9. Okay, so if you didn't know all your numbers, you might have thought it was that one. But the membrane attack complexes poke the holes in the membrane, actually the outer membrane. Peptidoglycan layer is really thin for gram-negative cells. It punches through that into the cell membrane. Okay, so a gram-positive cell with a really thick layer of peptidoglycan, it's not going to really do much. Okay, so especially acting on gram-negatives. I thought, since I have had a lot of questions about it, I would just bring up this slide again. This is actually a figure from our old textbook. A lot of questions about, well, what do I have to know about complement? Okay, we talked about the 3 different ways that complement is activated. One of the ways is when an antibody, you might recognize that as an IgM antibody now, but I don't think it necessarily has to be IgM. Recognizes an antigen, when the C1 complement protein recognizes an antigen antibody complex. Okay, that's one of the ways that complement can be activated. The other 2 ways involve the mannose sugar that's on a lot of bacterial cells that's not on our cells. That's recognized by proteins called lectins. That can split C3 complement fragment. Also, C3 is constantly being split a little bit on its own. Or again, the classical activation, any of those ways eventually act on C3 to split it. Okay, and that's kind of an important point, because after that step, now we're starting to get these fragments of proteins that are starting to actually act and help us fight invaders. Okay, that C3b acts as an opsonin. The C3a triggers inflammation, along with C5a also triggers inflammation. So that triggering chemotaxis and inflammation, those two things kind of go hand in hand. It's again the C5 and the C6, C7, C8, C9 that make the membrane attack complexes. All right, so remember, an opsonin is something that binds to do the pathogen. Okay, binds like C3b, binds to the pathogen and makes our phagocytic cells more likely to be able to grab a hold of that pathogen. Okay, yeah, question.
Do we need to know the mechanism why that happens, or just that?
So do you need to know the mechanism? Not really, just that I would think of like C3b as kind of like a handle for the pseudopod to be able to grab a hold of. So that's as far as I would go there. Any other questions about complements? Okay, it's one of those things you just kind of gotta read over a number of times to get it to sink in. Okay, question about cells. Which one of the following is mismatched? All right, let's go through them. B cells mature into plasma cells. Yes. Okay, I know that term plasma cells, that makes you think of blood plasma, maybe, but this is a certain kind of cell. I don't know why we call them that, but when B cells get activated, they mature into plasma cells. And remember, the plasma cells are the ones that make the antibody. Okay. So that's true. Monocytes mature into macrophages. Yes, that's true. They can also mature into what other kind of cell? Dendritic cells, good. This is the one that is not true. Okay, lymphocytes, they don't really mature into anything else. There are different kinds of lymphocytes, or different kinds of T cells. As far as I know, there's only one kind of B cell. But so yes, this is true. Myeloid stem cells mature into eosinophils. Oh yeah, right. Got that one. Okay. So this, you know, I can ask a lot of different questions. If you remember back to this drawing, this was, I'm trying to remember, this was last Monday's lecture, I think. So there's a lot of different things about the different types of cells. They differentiate, and remember, they start out as a stem cell in the blood, and they differentiate by a process called hematopoiesis into different kinds of stem cells, and then eventually into our granulocytes, which here the basophils, the neutrophils, and the eosinophils. Those are granulocytes. The agranulocytes are the lymphocytes as well as the monocytes. So that's what I circled there. Those are the main types of immune cells that we talked about. We did mention natural killer cells. Technically, those are lymphocytes as well. They kind of act in both innate and adaptive immunity. Okay, so kind of be familiar with that. Questions there? All right, so now we kind of get into our adaptive immunity. Remember, adaptive is just, you know, it's an artificial designation, right? Innate versus adaptive, but adaptive usually refers to responses that involve B cells and T cells, really specific responses. Okay, I'll give you a second to read this. I think this is a fun question. I have put it on exam before, but. Okay. What do you think? There's actually one answer that I thought, oh, that could be almost true also. To me, the best answer here is D. Okay, we talked about MHC1, MHC2. MHC1 is just a way for cells to display what's going on inside the cells. I think the word I used was volunteer or advertise. And only nucleated cells have that MHC class 1. So red blood cells, no nucleus, they don't display MHC class 1. So pathogens that invade red blood cells, how does our immune system notice them? It's problematic, right? Maybe that's one of the reasons that malaria persists as long as it does. Okay, the one that I thought, ooh, maybe that's true as well, and I don't think I made this question up. Red blood cells, they're not supposed to enter lymphoid tissue. I suppose maybe something could go wrong and they could eventually, but that's, you know, lymph is not red, it's white blood cells in there, but it shouldn't be red blood cells. So they don't get the appropriate signals perhaps to ever leave, even during inflammation, the red blood cells shouldn't be leaving our blood vessels. Okay, questions there? All right, T-dependent antigens. Read this one carefully. I'll give you a little time. Okay. So remember, a T-independent antigen is one that doesn't require, where the B cells don't require extra stimulation from T helper cells in order to be activated to differentiate into plasma cells and produce antibodies. Okay, so these are not as common, the T-independent antigens, but the ones that we have, we do understand our T-independent antigens are typically capsules, so polysaccharides from capsules. So the one that is not true is that they're usually proteins. Okay, all the T-dependent antigens are going to be proteins. Okay, so remember we said what's special about a T-independent response. Kids don't have a very good response to that. Their B cells just aren't very mature. This, I probably, you know, that's probably a little bit mean to throw in an example there. I always tell you don't need to know examples. So that might have been a little bit mean if I would have put that on the test, but we use that, that organism's capsule. I said that functions as a T-independent antigen. Again, this one is true as well, that the secondary immune response. So in other words, you know, the first time you get an immune response like that, and then the second time, it's about the same. You get the same kind of antibody, IgM. It doesn't switch into different kinds. With a T-dependent response, you get the secondary response. You get much more antibody faster. Remember, different kinds of antibody. It will switch. You'll have an IgM and then switch into IgG, for example. Okay. All right. Couple more. All right. Sometimes I'll let you read it and answer. Okay, remember you advertise or volunteer, one hand up, MHC class 1. Okay, so what kind of cells do MHC class 1 present antigen to? Cytotoxic T cells. Okay. MHC class 2, remember, this is just 3 kinds of cells, B cells, macrophages, and dendritic cells. Present what they have phagocytized, or in the case of B cells, kind of ingested, and they're going to show or present antigen that they have phagocytized, and they're going to present to T helper cells. Okay. Good. Okay, last one. Oh no, I got 2 more. Scratch that. I just gave the answer away. Forgot that I had this one. Okay, see, get it? It's a little nuance. Okay, so correct answer there should be E. I have had a number of people ask what's the difference between an exogenous antigen and an endogenous antigen. So an endogenous antigen is one that's inside our host cell, not inside a pathogen, but inside, endogenous meaning inside one of our cells. So a virus would start out exogenous, and once it gains entrance to our cell, now it's endogenous. Okay, so again, a different way that needs different ways the immune system detects an antigen, whether it's outside of the cell or inside of the cell. So anything that's phagocytized, okay, that's outside of the cell, and that's an exogenous antigen. Okay, and then, like I said before, MHC class 2 only will present to T helper cells are the class of cells that will recognize antigen presented by MHC class 1. Okay, so knowing that, that might make this last question a little bit easier. I'll let you read it. You have to kind of figure out what kind of cell that is and all those interactions being made. Don't worry about this and this. That's something we didn't talk about. So this is the kind of figure that, you know, if I don't think I had this exact one in your notes, but you would want to, it's kind of good practice to maybe draw it out on your own and see if you can label it yourself. The pictures should make something more clear rather than be complicate things, right? All right, so if we, knowing that this is a CD4 cluster of differentiation, okay, that tells us it's only the T helper cells that have CD4. Remember, it's the cytotoxic T cells that have CD8. And that CD4 binds to this MHC class 2. Okay, yeah, the antigen presenting cell, like a dendritic cell or macrophage, is presenting antigen. We could say some epitope of an antigen. And then this is the T cell receptor that's recognizing that antigen that's being presented by the MHC class 2. So knowing what all those parts are, again, that you could work out given the CD4, we're talking about activation of a T helper cell there. All right, so do you have other questions, other things from either a recorded video or class? Yeah.
Can we talk about toll-like
Can we
receptors?
talk about toll-like receptors? Okay. Let me just go back to that slide. All right, so toll-like receptors. So these are going to be present on any phagocytic cell. There's other kinds of cells actually that have toll-like receptors as well. They're a kind of pathogen recognition receptor. And their job is to recognize these broad-based patterns that we see in pathogens. So we have a toll-like receptor, TLR4, that recognizes LPS, for example. That's something that we find on the outer membrane of gram-negative cells, right? We have toll-like receptors that recognize peptidoglycan or flagellin. These are things that we never have on our cells, but it's not terribly specific. There's only like 10 different patterns that we've discovered so far that are recognized. Okay, so once a pathogen once a part of a pathogen is recognized with a toll-like receptor, that sends signals to the cell that yes, this is something that should be phagocytized. This is not self. It also will be kind of like, I think the analogy I used was like a burglar alarm. So it kind of trips this burglar alarm to send cytokine signals out to other cells, to call other cells there, to the area, to chemotax towards that pathogen. And it can just activate lots of other aspects of our immune response. Okay, so toll-like receptors recognize PAMPs, the pathogen-associated molecular patterns of pathogens. Yeah.
Can you just clarify what LPS is?
Yeah, so what's LPS? That stands for lipopolysaccharide. That's in the outer membrane of gram negatives. Okay, so the outside of the peptidoglycan layer. Part of that is lipid A, which is otherwise known as endotoxin. Okay, endotoxin is part of that lipopolysaccharide. Anything else you want to talk about, review? Okay. All right, don't be afraid to send me questions. Don't be afraid to stop by for office hours. I've been a little bit busier than usual, but not extremely so. All right. Okay, so as a good segue into talking about vaccines, we're going to start out by just kind of defining some terms that describes how one would get immunity to some infection. How would you acquire immunity to some infection? So in other words, how would we, usually we're thinking about the action of antibodies. Like, can we make our own antibodies? Do we get antibodies from somebody else? Can we get them artificially or naturally? Do we get them actively or passively? Okay, so those four terms, and then we squish that together. Okay, so the difference between an artificial acquisition of antibodies and a natural acquisition is basically, does it involve technology? Okay, so again, artificially involves some kind of technology, and natural, no technology. So I always ask myself, is this something that we could do 500 years ago? If yes, then it's natural. If it requires maybe a syringe or something like that, then that's going to be artificial. Active versus passive, somebody with active immunity, that means they are making their own antibodies. So they, have B cell activation, again, that's making their own antibodies. And if somebody has passive antibodies, that means they have preformed antibodies from someone else. From another person, or occasionally we will have a horse or something, inject a horse with something, and then harvest antibodies from an animal. Okay, so putting those terms together, some examples. Naturally acquired active immunity. Okay, that's when someone has the disease and then is immune following recovery. Okay, I had chickenpox when I was 8. I recovered from that, and that's one disease that we have really good naturally acquired active immunity from. Not that I've, every time I'm exposed to chickenpox, I make a good response to that, good enough that I don't even know that I've been exposed. I don't even get sick. Okay, so you have the infection, and then as a result, you have good immunity from that. Artificially acquired active immunity, that's when somebody gets a vaccine, and the vaccine causes us to make have a good immune response, good memory cells, so that the next time we're exposed to it, again, those memory cells kick in and we don't get sick. Naturally acquired passive immunity. So passive immunity, remember, we're just getting antibodies from someone else. And the only way that I can think of to do that naturally is breastfeeding. Okay, so in breast milk. Or then the last category, artificially acquired passive immunity. That's where we might get an infusion of antibodies. Okay, so sometimes if someone is, say, has some kind of genetic deficiency in a certain kind of antibody, like there are diseases where somebody can't make IgA antibodies, for example, they might go in every few months and get an infusion of IgA antibodies. After the Ebola epidemic, they tried treating people that were sick with Ebola with antibodies that they'd harvested from somebody that had recovered from Ebola. And it didn't really work out all that well. We tried doing that with COVID as well, it has some help. Okay, so those are the different ways that we acquire the ability to be immune through an infection. And a couple years ago, when the COVID vaccine first came out, I had this discussion with an acquaintance that I'm not totally proud of now, but she was a nurse and I sort of expected her to be logical, but we were having a discussion about vaccines. And she was very against getting the COVID vaccine, and her point was, she said, I just think eventually we'll all have antibodies to COVID. And after I thought about her saying that later, and I didn't say this at the time, these are the 4 ways that we become immune to something. Okay, either you have the infection and you get better, either you have a vaccine which protects you, either you get antibodies from somebody else, or you find somebody that is a lactating female that has recovered from COVID, and she lets you drink her milk. Okay, those are the 4 ways we get antibodies, right? You're not going to just breathe them in the air. All right, so anyway, that's a pertinent discussion. These are the ways we get antibodies. So as we go forward thinking about vaccines, it's not new to have people against vaccines. There's been an anti-vax society for as long as there's been vaccines. Okay, I didn't include this picture, but it's quite an interesting one. Of course, 1796, that's when Edward Jenner is remembered for using some pus from a milk cowpox lesion and sort of poking that pus with a knife under an 8-year-old boy's skin. And then 2 weeks later, he intentionally exposed that boy to smallpox. And he had some, he noticed that, oh, milkmaids tend to not get smallpox, probably because they've had cowpox and they seem to be immune. So he tested that out. It's not the way we do clinical trials today. And the boy didn't get smallpox. So there's evidence that maybe Edward Jenner wasn't the very first one to do that. I think they talk about that in the text a little bit, but Edward Jenner certainly was the first one to promote it and try to get other people to do it. And that was his life's work. So this picture was put out by the Anti-Vax Society. We're afraid of things that we don't understand, right? And even Edward Jenner didn't understand why people were protected from smallpox. But certainly, people were not throwing up cows or giving birth to cows or having cows coming out of some other orifice of their body. So again, we tend to be afraid of things that we don't understand. That's not new. Certainly the way they were administering this very first vaccine was dangerous. Like there was a huge risk that you could get some other, skin infection or a serious infection from the way that they were inoculating. Vaccines have gotten, they've only gotten safer, right? There's been some problems along the way, and we'll mention a few hiccups, but again, they're much safer today. That is a picture of what cowpox looks like. And that would be no picnic. But it's a pretty mild infection. It's supposed to be, and you don't ever die from that. So smallpox carries about a 30% mortality at best. So I guess it's better to get cowpox instead of smallpox. But yeah, it looks pretty nasty. Okay, so those two words, vaccination and immunization. Most of the time, people would use those two terms and we mean the same thing. Now, Aaron Rodgers, a couple years ago, I know some of you are Packers fans, so sorry to stomp on that. But he said, I've been immunized. Okay, what he meant by that was, I've taken some supplement, like some herbs that have jump-started my immune system. He didn't mean I am immune to this specific pathogen. When we say somebody has gotten a vaccine, that just means they've gotten a shot. What happens in our body is immunization. We get the B and T cells, the memory B and T cells as a result of that vaccination. So again, usually people mean the same thing. He took those terms literally and kind of, it wasn't the only one maybe that kind of misled people. But if you want to mince words, they are slightly different meaning. Why do we vaccinate? So we especially want to protect children. There's lots of infections that are much more serious when children get them. Okay, especially babies. So oftentimes, as soon as we can, there's some vaccines that we can't give to babies because it doesn't do any good. You have to wait a little bit. But we especially want to protect children. And then with the hope that immunity will last into adulthood. Older adults, we know that our immunity starts to wane once we get to be to a certain age. So there's lots of vaccines that you can encourage, older people that to get. Okay. And then, of course, the goal, we heard this term a lot during the pandemic. I don't think we're ever going to get there with COVID, but the goal is to have enough people vaccinated so that we have herd immunity. Okay, so that means that we want to, if most of the population is immune, then people that can't be vaccinated are protected. Okay, there's always reasons why some people can't be vaccinated. Maybe they have an immune disorder where it doesn't do them any good. Maybe they've had a strong allergy to a similar vaccination in the past. or maybe they're too young for it to do any good. So little babies, again, not everything can they be vaccinated for. And there are people that are just, again, medically fragile, that it's not a good idea for them to get vaccinated. So the idea is that we try to vaccinate everyone else so that our herd immunity is high enough so that, there's so few people that are susceptible to the infection that it just doesn't really have a chance to pass. It doesn't have enough susceptible individuals. Okay, so I think, to me, this is the biggest reason to get vaccinated. Some people would say, well, it's my body, it's my choice. But if you get sick, and I'm not just talking about COVID, I'm talking about other things, we can mathematically kind of predict how many other people that you would get sick. Okay, so one infected individual for influenza, for instance, is likely to get between 1 and 2 other people sick. Okay, so if you say, well, I'm not going to be vaccinated, and you get sick, then you're likely to get a couple other people sick. So it's not just an individual choice, and I'm not saying that we need to make everybody definitely get vaccinated, but there is certainly an effect that you have on other people. Okay, so of course the goal of vaccination is that we want to induce adaptive immunity. We want to get memory B and T cells. Okay, so the next time that we're exposed, we don't have to make all those different immunological synapses, and we get a very fast response. All right, so I'd like to go through and talk through the most common types of vaccines that we have out there. This is a lecture that I've had to expand on quite a bit in the last few years as we've gotten lots of work done, different types of vaccines that we're using. The first vaccines that we had, including Edward Jenner's vaccine to smallpox, have been attenuated vaccines. So that just means that the pathogen is weakened in some way. That's what attenuated means. So it's live. And how have we weakened that pathogen, whether it's a virus or a bacteria? Sometimes we grow it in a cell type that's not its normal host. For example, the attenuated polio vaccine is grown in monkey kidney cells instead of human nerve cells. Okay, so sometimes we grow it in a different kind of cell type. Sometimes we grow it at a higher or lower temperature, or just for lots and lots of generations, and it kind of loses its virulence. Okay, sometimes we don't really exactly know what's changed, like the tuberculosis vaccine. We don't really know exactly what changed from the original wild type, but it seems to work fairly well. So a few advantages, and then we'll get to the disadvantages of this type of vaccine. It's live, so you get a very natural immune response. If it's a virus, it enters the host cells, and that virus gets presented by MHC class 1. We get good B cell, good T cell activation from that very natural adaptive response. So usually fewer doses are necessary than what we see for the killed vaccines or the toxoid vaccines. And this can be an advantage, it can be a disadvantage, but if it makes people, it actually gives people a mild form of the disease, they may actually be infectious and can give that mild form of disease to other people. So we call that contact immunity. So even people that didn't actually get the vaccine might be able to get immunity if it forms a mild form of disease. So that might be an advantage if you're trying to reach a remote population, maybe a remote village. And if you can at least get half the village vaccinated and they're all living together, the ones that weren't vaccinated can acquire immunity as well. Now, today we would actually, in our society, we would generally consider that a disadvantage. So I'm going to write that here. Possible to have transmission of the pathogen. Okay, we don't want to pass on even a mild form of the disease, because there's a chance that we could pass it on to somebody that was, immunologically immunocompromised, or pass it on to a newborn baby that has maybe not, not as robust an immune response. So generally, at least in our society, we don't want to do that. We don't want contact transmission. It's a little bit more difficult to work with these vaccines because you have to refrigerate them. They're live organisms. So the costs with transporting them are going to be higher. What's the third thing? Oh, the big one is that although rare, there have been instances where reversion is possible. Okay, and what I mean is reversion back to the wild type or infectious type. Okay, so when this happens, so a good example here, we actually have two different vaccines for polio. We had the Salk vaccine, SALK vaccine, that came out first. That was it's actually a subunit vaccine. We'll talk about those in a minute. And then a few years later, Sabin came out with his vaccine that was an attenuated vaccine, and it worked much better. So we switched in this country to the Sabin polio vaccine. Well, and that was the vaccine, it was oral. That was another nice thing about it. So it came in these little vials and you just had to drink the vaccine. So, no syringes, no pokes required. You just get your baby to drink this little, sweetened vaccine. Great, it worked great. The problem was in the year 2000 in the Dominican Republic, we had about 20 kids that got polio from that Sabin vaccine. Full-blown polio. So the mutated form had reverted back to the wild type and they got polio. So ever since 2000, we don't use that Sabin polio vaccine in this country anymore. It's still used other places worldwide because, again, the chances of this are really, really low. But we don't use it anymore in this country. Okay, we went back to using the Salk one. And because polio is so rare, polio is only transmitting, there's only natural transmission of polio in a few countries, Nigeria, Pakistan, and Afghanistan. We're really close to eradicating polio. So we still do give the polio vaccine to infants in this country, but hopefully we won't have to do that for much longer. In other countries where they're still giving this attenuated Sabin vaccine, that virus, the polio virus, is excreted in the feces. So it ends up in untreated waterways. And there have been cases of people getting polio where it's reverted in the water. I think there was a case in New York City not that long ago where somebody actually got polio from, again, being in some untreated water somewhere. So again, if we could stop giving the vaccine altogether, if we could eradicate it, okay, then that's the goal. Okay, but we still do use some attenuated vaccines. Usually we only use an attenuated vaccine unless we can't get any other type of vaccine to work. So we had a measles vaccine back like in the 50s or 60s. It didn't work very well. And so we had to go with the attenuated, MMR. We put the measles together with mumps and rubella, yellow fever, some influenza. So there's a certain age group that it works a little bit better to get an attenuated influenza virus. I think you breathe it in and it only infects places of your body that are a little bit cooler, like your nose. Chickenpox virus is also an attenuated one that I didn't list there. Okay, so don't memorize the examples. just to kind of tie it in. Probably the most common kind of vaccine, we call them killed vaccines. Are viruses alive? No, so maybe it's better to say inactivated if it's a virus. It can either be a whole cell that's killed or just a subunit, just part of that pathogen. We usually pick a very, something on the surface of the pathogen that our immune system recognizes really easily. Okay, so killed vaccines, okay, they are tested every batch. There's quality control and tested to make sure, however they killed it, that yes, that pathogen or virus was inactivated or killed. In general, more boosters are required because, again, we just don't get quite as natural invasion of cells. We get mostly a humoral response. Again, so we get mostly, we get good antibody activation. We get less activation of those cytotoxic T cells because, again, that killed pathogen doesn't get into our host cells as easily. Okay, so there's some examples there. Again, that killed can either be a whole cell or a subunit. How do we kill it if it's a whole cell? Usually treated with some chemical, perhaps. Sometimes radiation is used. Sometimes just high temperature will kill it. The main problem here, I don't know, I'm not sure if there are any whole cell vaccines in use right now. The one that's kind of famous that we switched lately is the vaccine to pertussis, also known as whooping cough. And this is pertussis is caused by a gram-negative pathogen. I know we haven't talked about that yet. Hopefully we'll get to in the next few weeks. Inflammation was actually quite a big problem with the pertussis vaccine. We typically give pertussis along, that D stands for diphtheria and the T stands for tetanus. Okay, so P is that whole cell pertussis. So, I mean, there's no doubt that really cut down on the number of pertussis infections. In the 30s, before we came out with that vaccine, we had over 200,000 cases of whooping cough in the US per year. With the vaccine, 1976, we were down to like 1,000 cases. We started to kind of increase in the early 80s, and it's not really clear why. Still kind of on the uptick today, more so because we actually switched our vaccine. So now we actually do this, we give it DTAP. Okay, that was really sloppy. Let me write that again. DT. Okay, so we still give it pertussis along with diphtheria and tetanus, but the AC stands for acellular pertussis. Because that pertussis vaccine caused a lot of inflammation. So things like redness, pain at the site of infection, maybe a little fever. You know, not a big deal, but you know, when mothers are vaccinated, getting their babies vaccinated, and they're really fussy and cranky after that, then it maybe they're going to decide not to go back for that next dose. So they figured out that what was causing the inflammation is that endotoxin that's part of the outer membrane of all gram-negative pathogens. When those cells are dead, the outer kind of envelope or cell wall starts to break down, but there's enough of it present still that lots of inflammation resulted. So they switched from using a whole cell to just parts of that. They took certain parts of that pertussis bacterium, and now inflammation is much less. Okay, so there's lots of different, if you look up this vaccine, there's lots of different varieties with capital letters and lowercase letters. It's kind of different, different ages get different kind of variation of that DTaP vaccine. But when it's the AP, it's always the acellular pertussis. Okay, and then a subunit vaccine, again, more boosters because we're only taking a part of the pathogen, not as many epitopes. So sometimes it takes a while to really teach your immune system to make those memory cells, but less problem with inflammation than some whole cell vaccines. Oh yeah, I wrote that down there, I forgot. Okay. So again, we have a lot of subunit vaccines. A toxoid vaccine, there are some infections where the disease is due to maybe a particular exotoxin that the bacterium produces. We don't have vaccines to endotoxin. We haven't been able to develop that, but exotoxin, yes. We just inactivate the toxin again, maybe by heat or some chemical. We get a really good antibody response, which is usually enough. Remember, those antibodies will bind to it to neutralize the toxin. So tetanus and diphtheria are the two vaccines that are toxoid vaccines. And actually, that pertussis vaccine, we actually, part of the fragment that we use is from one of the pertussis toxins. So you could sort of consider that kind of a toxoid vaccine as well. Diphtheria is an infection that we won't get to later on. Before we came out with the diphtheria vaccine, 200,000 cases annually. Now there's less than one in the US every year. So when you're infected, you get this sort of pseudomembrane in your throat that can actually suffocate you, and lots of different complications from that. So this was really a bad infection in children, especially. Tetanus, we will talk a little bit more about, hopefully. You know, the tetanus vaccine only lasts maybe 8 to 10 years. Why do we lose our immunity to that? Probably it's just that toxin is just a smaller molecule. There's less epitopes. We just get a less memory to it, I guess. So that may be a reason. We don't really know, but that may be a reason that you have to get that renewed every so often. Okay. All right. So a few other kinds of vaccines. Conjugate vaccines, really you could consider this a type of subunit. It's taking a part of a bacterial cell and actually part of the capsule. Okay, so they take, and we talked about how those polysaccharides are not as antigenic. They're not as good at eliciting a strong immune response. So what we do is we conjugate or attach part of those polysaccharides from the capsules and attach them to some protein that the immune system is much more likely to recognize. And remember, we talked about polysaccharides. Some of them are those T-independent antigens. Well, not all of them, but some of them. So, okay, when we attach that to a protein, we can get even a better immune response. Okay, so some examples. This is an old picture, Prevnar 13. That vaccine is actually, anybody know what that vaccinates against? Okay, Streptococcus pneumoniae. Sometimes we refer to that as pneumococcal pneumonia. Lots of different kinds of pneumonia. And that's, again, I think the updated version of that is Prevnar. It's either 20 or 23. I'm not really sure. There's a couple of different variations of that vaccine, but both of them are the Prevnar 13 was a vaccine to 13 different variations of that capsule of that Streptococcus, and now they've updated it to recognize more variations that are possible. And again, there's a couple different kinds of vaccine depending on how the age, if you're inoculating babies or older adults. But it's drastically reduced the incidence of a very severe kind of pneumonia, as well as some other things like sinus infections and earaches. Neisseria meningitis. It's a hard one to spell. Okay, we're going to talk about the Neisseria hopefully in a couple of weeks. Sometimes that's called meningococcal pneumonia. And we'll talk about that vaccine, but again, it's a vaccine to the capsule. The other one that we talked about is the Haemophilus influenza, that capsule, which is a T-independent antigen along with the Neisseria meningitis. So, okay. And then I want to at least mention the COVID vaccines. Of course, there's a lot of resistance to messenger RNA, that COVID vaccine when it first came out because it was new. But we've been doing research on messenger RNA applications for 30 years. They were at first hoping to use them as drugs, but then it became apparent that maybe it would work a little bit better to use them as vaccines. A lot of potential to use messenger RNA, not only for vaccines against infectious agents, but also against cancer. So there's a lot of work being done with that. Advantages, disadvantages of this kind of vaccine, it's very safe. There's in terms of, there's no way that a person is going to get the infection that you're vaccinating against from the vaccine. That would be impossible. It can't mutate your DNA either. And it's very quick and fairly inexpensive to produce them. The influenza vaccine that we use, it takes like 6 months to get a large enough quantity. So we have to kind of start preparing for the flu season, like the February before. With mRNA vaccines, we can make them very quickly, make lots of them very quickly. Disadvantages, that messenger RNA, it's a very fragile molecule. It degrades really quickly. So we have to keep the vaccine super cold so it doesn't degrade. Some short-term inflammatory reactions are pretty common. They've had to really monkey with some things. They've had to kind of modify some of the nucleosides a little bit because those viral nucleosides were being recognized by our toll-like receptors and causing a lot of innate immune response. We don't want an innate immune response. We just want the memory cells. So we're trying to figure out how to dampen that. And this myocarditis, that just means an inflammation of the heart muscle. Mostly would cause just a little bit of chest pain. They found that about 1 in 50,000 people would experience that after their second dose of one of the mRNA vaccines. They found that they can make that happen less often if they increase the time between the first and second dose. So when those COVID vaccines first came out, they were in a hurry to get everybody vaccinated, but now they'll make a little bit more time between the first and second dose. Incidentally, if you get COVID, the chances are about 3 times greater than that you'll get myocarditis. So you gotta always weigh your risks. I just mentioned quickly DNA vaccines. Of course, that Johnson & Johnson vaccine, if you'll remember, is a DNA vaccine that is also considered a viral vector vaccine. So they would put the DNA that encoded the spike protein of COVID into a virus. And that way it could be delivered into our cells. It was a virus that couldn't replicate in our body. It was taken from a chimpanzee. We're not using the Johnson & Johnson vaccine anymore unless somebody can't tolerate any other kind because of these rare side effects that were serious, that a few people died from, this clotting disorder, this thrombocytosis, a little bit higher incidence of this Guillain-Barré, which can result from any viral infection, but it's kind of a paralytic, usually wears off. Students or people really usually recover from it, but a little bit higher incidence of some more serious side effects. So for that reason, again, not using that now. Lastly, I'm not going to go in as much detail about this vaccine either, but if you think about, I always used to tell students, well, there's of course lots of infections that we don't have vaccines for. And that doesn't mean we're not working on it, but a lot of times the reason we don't have vaccines for something is because the pathogen changes too quickly. And that was the case for malaria, that Plasmodium protozoa. There's just so many variations of this that a few years ago, I used to say, they don't think it's even possible to have a vaccine for that. Well, lo and behold, 2 years ago, there's one that's been approved by the FDA. So we do actually have a vaccine to malaria that's mostly given to kids. It's called Mosquirix. It's kind of the name that's a little bit easier to remember. It's also called RTSS. But it's a recombinant vaccine. So that just means we've used molecular biology techniques to like take this piece of the pathogen, put it all together and actually add something to it. It's actually some protein from hepatitis virus to make it more immunogenic, more easily noticed by our immune system. So I won't go into the details of that. But the idea is that, again, we give this to kids and then it keeps, it tries to keep the protozoa from infecting the liver. Remember, kind of general life cycle that protozoa Plasmodium infects the liver first, matures there, and then goes to infect the red blood cells. So it tries to keep it from even getting into our hepatocytes, our liver cells. It takes a lot of injections. So 4 injections is kind of hard to do. Remote places of Africa where malaria is most prevalent. And interestingly enough, it only has about a 30% efficacy in preventing severe malarial disease. So most of the time, a vaccine would not be approved unless it's, works really well, somewhere above 50%. But in this case, there's about 1,000, 1,200 kids a day that die of severe malaria. So if they can keep a third of those kids from getting malaria severe enough that kills them, then it's worth it. Okay, so as of last year, they had given this as a kind of trial to a lot of kids. There's a lot of hope that we're going to keep making this better and better, and that it will save a lot of lives. Okay, so skip over that. Okay, so as we kind of wrap up here, it's important to think about, when COVID vaccines were pulled out, a lot of people said it's too quick. How do we know that they're safe? Well, you should sort of be aware this. I pulled this off the CDC website of, how are vaccines approved in the US? Well, even before they get to clinical trials in humans, there are extensive testing done in animals. Okay, so sometimes we don't have a very good animal model for a disease, but at least we can tell if that vaccine is harming animals. Okay, so lots of observation and testing in animals before we even get to a phase 1 clinical trial. And in phase 1, you're taking a small number, somewhere between 20 and 100 healthy volunteers. So people that don't have any other chronic problems, and they're just seeing the effects that it has on them. Is it causing any side effects? Is it working in preventing the disease? Sometimes, again, with a small number of people, that's hard to tell. If everything looks good, the data looks good, usually side effects show up within a couple of weeks. They wait longer than that. And then in phase 2, they're giving it to more people, and not just healthy people. They're varying the dose to see what works right. They're giving some people a placebo. Okay, they're controlling it. So you've got people that are just getting saline or something injected into them. And then they wait, they get the data together, wait a certain period of time, and before they go into phase 3, where they're giving it to usually thousands of people. And again, collecting data, see if it's keeping them from getting sick and if it's otherwise safe. So when they did this with COVID, they did kind of rush the time between phase 2 and phase 3 a little bit more than what they normally would, because it was considered a national emergency. But otherwise, they went through all those stages. And then an independent advisory council, not the manufacturer of the vaccine, but an independent group looks at the data and decides whether it should be approved. Okay, so the FDA will take, the input from this advisory council, and the FDA will decide to approve it or disapprove it. And then after that, there's really a stage 4. We call that the VARS stage. That stands for Vaccine Adverse Effects. What does the R stand for? I'm blanking on that. Reporting system. Okay, so normally, up until last few years, this would be, if somebody had a problem with a vaccine, they would call their doctor, and that was the reporting system. Now, I know when I got my COVID vaccine, I opted in to getting text messages, and periodically this VARS reporting system would say, how are you feeling today? Had any adverse effects from your vaccine? Are you generally in good health? Have you gotten COVID since the vaccine? So I had a chance just to be a data point to see, if the vaccine was, generally working and having any adverse effects. Okay, and that's just ongoing, like forever after. I mean, hopefully I won't get texts forever, but at any time, there's that can be reported and that information. Okay. So again, to wrap up, we can't really argue with the data that vaccines have certainly prevented a huge amount of infectious disease. If you look at a couple of them, polio and measles, at the time after we introduced the vaccines here, incidence went way, way down. Incidence and prevalence. Remember, incidence is the number of people that newly got the disease. Prevalence is the number of people that are kind of living with it for a certain amount of time. Measles is so infectious that pretty much before vaccines, everybody just got measles, and there were some pretty nasty complications that could happen as a result of that. So now after vaccine, it's very low. Back in the late 80s, they decided to start giving it in 3 doses. So once you maybe graduated from high school, they recommended that you get another booster. And after that, now that everybody has 3 measles vaccines, incidence is really quite low. Okay, so again, however you feel about vaccines, whether you want them or not, you can't argue with the fact there's a lot of childhood illnesses that we don't have to deal with anymore. Okay, any questions, comments, anything about vaccines? Okay, don't pack up yet though. I'm using every minute. Okay, I do want to go on and start our first pathogen. Got 5 minutes left. Again, Staphylococcus, and this is familiar. A lot of you starting your unknowns in labs, so this might, hopefully reinforce what you're learning in lab. But I want to talk about a couple different medically interesting gram-positive cocci. And I should mention that after our next exam, either Wednesday or the following Monday, I'm going to talk a lot about what I hope to cover in the next few weeks in terms of which pathogens and what the final will look like. A number of you have asked me that already. So more information on that going forward. But this is the time in the semester where we just pick certain pathogens and we like to talk about it. And hopefully there's things from the rest of the semester that we can kind of tie in, and hopefully things kind of all start to make sense. So there's a couple of gram-positive cocci. Your book covers this pathogen information coming from more of a systems perspective. So it will say, oh, infections of the skin, infections of the respiratory system. And I tend to like to talk about an organism rather than a system. So there's a little difference in the way I'll present it and the way your book does. But the Staphylococcus and the Streptococcus are two gram-positive cocci that we'll talk about. Enterococcus are very closely related to the Streptococcus. Okay. Staphylococcus. So there's how you count the number of species depends on whether you're talking about only ones that infect humans or ones that infect animals. So there's somewhere between like 20 and 40 different species of Staphylococcus. They all will grow in grape-like clusters. So remember your cell arrangements. We talked about this a long time ago. Diplococci. I looked at a few gram stains this morning that looks like they were pairing up. Okay, some organisms will do that, more so the Streptococci. Okay, Streptococci will grow in these long chains. And then sometimes you get the tetrads or the Sarcina. The organisms that do that are not famous ones, to be honest, but the Staphylococcus, again, will divide in many different planes, and then they stay stuck together in these grape-like clusters. Okay. Where do we find staph? They are just kind of common microbiota, especially in the skin, the upper respiratory tract, the mucous membranes. They're fairly common in nature. So things we touch, we tend to, anything we touch could have some staph on it. So you find them a lot of places. They are catalase positive. All right, so again, if you're in lab, this should look familiar. A catalase test, we add some hydrogen peroxide onto the organism, and if it makes catalase, we're going to end up with water and oxygen. So those bubbles that we're looking for, those are oxygen bubbles. Okay, so that's the way that we tell the difference between Streptococcus and Staphylococcus. Those look, very similar. They're both gram-positive cocci. Some form capsules, or some kind of capsules, maybe a slime layer, a less rigid capsule. They are facultative anaerobes. Okay, so flexible in their metabolism, and they are kind of known for being salt tolerant. Okay, an organism that lives on your skin can be kind of a sweaty place. Sweat is very high in salt. They're not halophiles. They don't grow best in high salt, but they tolerate it a lot better than other organisms, and in generally a little bit more environmentally tough than some organisms. So they tolerate temperature swings, for example, or pH swings much better than the Streptococcus. Anyway, streps are kind of wimpy. Okay. So we're going to look, we're going to pick one staph and talk much more about it because it's kind of the most interesting one clinically, and that's Staphylococcus aureus. I guess that's a good place to stop for today. So we'll probably start there Wednesday after the exam. And again, see me, lots of office hours. Have a good day.